Okay, so our next unit is on DNA and protein synthesis. And this is the structure of the DNA molecule. The DNA molecule is found in what we call the chromatin when we're talking about mitosis, in the chromosomes. The DNA, they just make up those chromosomes. So the DNA is a double helix. It looks like a twisted ladder. And this structure was discovered by Mr. Watson and Mr. Crick. So Watson and Crick discovered the double helix, twisted ladder structure of the DNA molecule. The genetic code. The genetic code is the way in which cells store the program that they pass from one generation to the next. It's how you get your traits from your parents. And that genetic code, that's in those four bases that we talked about, the G's and the T's and the C's and the A's. And we'll talk a little bit more about that in a few minutes. That's the genetic code, is how traits are passed from one generation to the next. Okay. So the structure of the DNA consists of a sugar called deoxyribose, a phosphate group, and a base. This is that nucleotide. The nucleotide that we talked about when we were talking about the macromolecules. So it is a nucleotide. The nucleotide is the building block of DNA. And that's back to that structure that we showed when we talked about those macromolecules. We had the sugars that we represented like that. And we do that because it has kind of that structure with carbons in those. Then we have what we call our bases that we represented usually with a box. That's our base. And again, that base is going to be either the adenine, the thymine, the guanine, or the cytosine, the A, T's, G's, and the C's that we talked about. That's our bases. And then we have the phosphate group over here that we represented as a circle most of the time. That's our phosphate. So we have our sugar. And since this is DNA we're talking about, that sugar is deoxyribose. And again, ose indicates that it's a sugar. Your bases are adenine, thymine, guanine, and cytosine, and this is our phosphate group. And that is the nucleotide that is the building block of the DNA. And it's the order of these bases that we're going to see in a few minutes that's going to determine what that DNA is coding for. So that's the nucleotide, the building block of the DNA structure. And there we go. DNA contains adenine that we're going to represent with A, guanine that we represent with G, cytosine that we represent with C, and thymine that we represent with T. And the thymine and cytosine belong to a group of compounds that we call pyrimidines, and we're going to look at their structures in just a second. Adenine and guanine go together. We call them purines. So you're always going to have a purine bond with a pyrimidine, and this purine bond with this pyrimidine. Okay? So adenine is always going to bind with thymine, and guanine is always going to combine with cytosine. So adenine to thymine and guanine to cytosine. Every time a purine to a pyrimidine. And there's what they look like. The pyrimidines, that's our thymine and our cytosine, they just have a single ring. Single ring pyrimidine, single ring pyrimidine, single ring pyrimidine. Then we have our purines. Purines are double rings. They have like a pentagon and a hexagon together. That's our adenine and our guanine. So our adenines are going to combine every time with our thymine, and our guanines are going to combine every time with our cytosines. So DNA is composed of two long chains of nucleotides arranged in a spiral. We call it a double helix. A double helix structure. It looks like a twisted ladder. And these rungs here, we're going to see in just a few minutes, these rungs are actually going to be the bases. This is maybe an adenine here that's sticking to a thymine here. And then our sugars and our phosphates go up the sides of our double helix. So sugar and phosphate make the sides, or what we call the backbone. And then our bases are sticking together here in the middle, held together by hydrogen bonds. So the sides of the double helix are made up of alternating molecules of sugars and phosphates. Sugars and phosphates are held together by covalent bonds. That's important to know. Sugars and phosphates are held together by covalent bonds. They're held tightly together. Covalent bonds are stronger. The rungs are the bases, the adenine sticking to the thymine and the cytosine sticking to the guanine. They're held together by hydrogen bonds. And that's going to be super important because those hydrogen bonds are weak bonds. They're going to break apart easily. That's when we get ready to do DNA replication in a few minutes, or when we open up the DNA to make mRNA, when we start talking about protein synthesis. It's going to be important that we're able to separate those bases and break that hydrogen bond easy. There's going to be an enzyme that will come in and break those hydrogen bonds so that we can get to them to replicate. Okay. So the four bases are said to be complementary because they go together. Complementary means the bases on one strand determines the bases on the other strand. So our DNA template that we're going to use to make our complementary strand, our DNA template, those bases, if it's an adenine, it's going to make it be a thymine on the other side. If it's a cytosine, it's going to make it be a guanine on the other side. If it's a guanine, it's going to make it be a cytosine on the other side. It's going to determine what happens on that other side. Okay. And what I was saying earlier about that backbone is that we will have our sugar, and then that base that we were talking about, and then our phosphate coming up right here. Well, this phosphate is going to stick to the bottom of that next one, that next sugar molecule, and this is how they're going to stick together to make up those sides. And then it's going to have another phosphate that will join to the next one, and then down here will be another one, join to the next one, and that's how it's going to make up the backbone. And then we have our bases coming off the side. So if this is an adenine here, then our next strand is going to be that weak hydrogen bond right there. This is going to stick to the base on the strand on the other side. And that would be a thymine. If this is an A, that's going to be a T. Maybe this is a C. This is a C. This one's going to be a G. And if this one's a G, this one would be a C. And these would be the bases of the other side, the complementary side. And it would stick to this one over here. And then we would have that sugar right there that one was stuck to. And that would have a phosphate come off there. And it would be stuck to the phosphate of the next one. And then this one would be stuck on that next one. And then it would stick to that phosphate. And we have a phosphate here. And so on. And these would be our rungs. It's sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Those are our sides of our DNA. And then our rungs are where our A's and T's and G's and C's and C's and G's and T's and H's, where those are all sticking together. That's the rungs. And this would be our hydrogen bond right there. It's our hydrogen bonds. And they're weak bonds holding the two sides of the DNA molecule together. Practice problems. If we have one strand of DNA, if we have an A, then it's going to stick to the T. A is going to stick to the T. A T is going to stick to an A. A is going to stick to a T. Okay. G, C to G, T to A, T to A to T, and G to C. And that would be our basis. This would be the middle of the DNA molecule held together by a hydrogen bond. You can see from the model, the structure is basically a rope lettering type thingy that's all twisted around. So the structure of the DNA in 1953, Watson and Crick discovered that twisty letter structure. And then there are James Watson and Francis Crick. So this is James Watson, this is Francis Crick. And in 1953, they were able to discover the shape of a DNA molecule. Functions of DNA. DNA determines which protein will be produced by a cell. The type of protein determines the activity in the physical fission cell. We talked a lot about proteins when we talked about the macromolecules. We said proteins are what give you your eye color, your hair color. They determine the shape of the hemoglobin in your blood cells. They determine whether you're type A blood, type B blood, type O blood, because it's the proteins on the surface of those blood cells. Every function in your body is pretty much controlled by a protein. And those proteins are made from the DNA. And the DNA codes those bases on the DNA as we get further into protein synthesis. So the order of those bases is what's going to determine the order of those 20 amino acids that we talked about. It's going to determine the order those amino acids are laid down. And the order of those amino acids determines the shape of the protein. And the shape of that protein is going to be what determines the function of the protein. So the function of the protein determines what proteins will be made by the cell. The type of the protein determines the activity and physical features of that cell. So the type of the proteins determine the activities and physical features of the cell. The sequence of those amino acids, that adenine, guanine, thymine, are the 20 amino acids that we see. Everything in the universe is made of the same 20 amino acids. Your oak tree has the same amino acids as the every protein is made of the 20 amino acids. You're made of the same 20 amino acids that your dog's proteins are made of, that the oak tree's proteins are made of. They're all the same 20 amino acids. The amino acid sequence is determined by the sequence of those adenines, guanines, thymines, and cytosines. So a series of 3 nucleotides, or 3 bases. Each nucleotide has a base on it. So a series of the 3 bases. It's called a triplet. It's called a triplet, and a triplet is going to be very important because each of those triplets is going to code for amino acids. So one triplet is going to give us one of those amino acids. One triplet is going to equal one amino acid. For example, ATT would code for amino acid. GCG would code for an amino acid. TAG would code for an amino acid. This right here, this is what's called a codon chart, and this is called a codon wheel. And how it works is we would look at a piece of DNA, and if it had bases A and then a T and then a T, well, that ATT would code for, we would go to A. Eventually, it would code for mRNA. It would code for mRNA, messenger RNA. It's going to take it, and then the ATT would code for mRNA. Then we would take our mRNA. We're going to use our mRNA here to tell us what amino acid we have. But this ATT would code for mRNA. We would take to our charts, or our wheel, and it would tell us which amino acid that this ATT, way back in the DNA, was coding for. Therefore, the sequence of the bases in the DNA makes up the code for a specific amino acid. So the sequence of the bases tells us the specific amino acids in a protein chain. When a cell divides, it makes new cells. Each new cell must contain the complete set of genetic instructions. We just went through a unit on mitosis, and in that unit of mitosis, we saw that the chromosomes are going to be pulled apart, and you're going to end up with 2 identical cells. Well, that means that that DNA had to be copied exactly in the first cell to make enough DNA from 2 cells to have the exact same thing. So a complete set of genetic information, exact copy of the DNA molecule. The DNA code is copied in a process called replication. That's going to be important to know. So DNA code is copied in a process of replication. DNA replication is copying the genetic code from one strand of DNA to another strand of DNA so that the cell can reproduce. So how does replication work? DNA replication is carried out by a series of enzymes called helicases. And we said earlier that deoxyribose was a sugar because it ended in ose. Well, helicase, ase, remember, means enzymes, which is a protein. So DNA, the enzyme is called DNA helicase and DNA polymerase. Those are 2 enzymes that come in and they open up that ladder up that middle where we said those weak hydrogen bonds are between those bases. Well, that's going to open up. It's going to kind of unzip it. So DNA helicase comes in and unzips the 2 strands of the double helix. And the DNA polymerase inserts the appropriate bases to produce the sugar phosphate link that extend the growing protein chain. So we're going to make another sugar phosphate backbone by connecting the bases. New bases are coming in. It's split it open. You actually have it happening on two strands at the same time. And let's see, right here's a picture that kind of shows what's happening. In DNA replication, the helicase comes in and unzips, and there's the representation of the helicase. And the polymerase is going to come in and kind of proofread and make sure it's going correctly. And this was our original DNA strand, and it's opening up. And as it's opening up, then these molecules, this DNA polymerase is coming along. This is the DNA polymerase. It's coming along and says, oh, there's an A, let's stick a T. There's a G, let's stick a C. There's a T, let's stick an A. So it's the whether or not this is an A or a G or a T or a C on this strand is going to determine what's going to be laid down on that next strand. And that's how replication happens. It's copying, so it's going to end up with this strand looking like this strand, and this strand looking like that strand on the original DNA molecule. Okay, so the helicase comes in, the DNA helicase comes in and it zips, and then DNA polymerase comes in and it's going to match up those complementary base pairs. It's going to match the T's to the A's and the G's to the C's. Polymerase proofreads. Polymerase proofreads. Polymerase proofreads. Polymerase is going to proofread and make sure those bases are put down in the correct order to match that DNA template of the original strand. Polymerase proofreads. When that goes together. And there's another picture of how it's happening. The helicase, DNA helicase comes in and zips, and there we see there's a T, the polymerase matched it with an A. There's an A, the polymerase matched it with a T. There's a G, the polymerase matched it with a C. And here we go, A to T, C to G, T to A to T, G to C. And they just kind of fit together that way. This was our original strand. DNA helicase came in and opened it up. Put that in there. DNA helicase. And zips. And then here, this A here, got a T. That T that was already on that side, it's going to get an A. And this is going to be this is the original, the dark blue is the original strand. And now the polymerase is coming in, laying down these strands. So then we're going to end up with two identical sets. This is an AT and this is an AT. This was our first A and our first T. And we added these on, so now we have two complete ones. Here, DNA polymerase. DNA polymerase lays down the bases and proofreads. Helicase and zips, DNA polymerase proofreads. Okay, so when the DNA in a cell replicates, the cell has two exact copies. Two exact copies. And that's important when we're talking about mitosis. You end up with two identical cells, so they have two exact copies of the DNA molecule. The cell then divides with each cell getting a copy of the DNA molecule. So why is replication so important? DNA replication is necessary to life because DNA contains the genetic information needed to form and control the physical makeup and chemical processes of an organism. Okay, so it's important that every cell have the correct order of those bases to be able to make the correct DNA molecule, to be able to make the correct proteins, because it's those proteins that are going to control everything in the cell and everything in the body. And that's example problems. We're going to write the complementary strand. This is our DNA strand. A goes with T. A is going to go with T. G is going to go with C. And we just got hydrogen bond in there. G is going to go with C. C is going to go with G. T is going to go with an A. T is going to go with an A. T. G is going to go with C. C is going to go with G. G is going to go with C. So that would be our complementary strand. Always, A bonds with T, and C bonds with G. And I remember that because C and G look a whole lot alike. It's just got this little tail thing on G. So C and G go together because they're so much alike. A goes with T and G goes with C. Quiz time. The primary function of DNA is to store information for building proteins is your correct answer. Number 2, the structure of DNA was discovered in 1953 by that would be A, Watson and Crick. What type of bonds hold the nitrogenous bases together in a DNA molecule? Those bases in the middle, what holds them together? Correct answer is A, hydrogen bonds, because we need a weak bond for DNA helicase to be able to open it up for replication. So hydrogen bonds. Cytosine and thymine are what is the shape of the DNA molecule? That would be the double helix.